So I'm going to come back later and do the plates that you guys have all been looking at, and we've kind of had some discussions one-on-one at the end. And we'll also talk about some of the other show and tell stuff then. Because as we go through this, I want to reiterate, and that's one thing Jack said is we play off each other. And that is something that has never been more true than these live events. Because there will be things that I learned from the speakers and I learned from you. And then you'll watch our talks will actually evolve as the day goes on. It's a pretty amazing thing, especially if you were to sit down and talk to Dr. Pompa, Dr. Barston, myself, Jack, we actually gain and modify and evolve throughout the weekend. And at Sunshine, it's even more pronounced because we learn off each other. I spend time with you guys, and it triggers new memories, it triggers new examples, new clinical cases. So it's an amazing phenomenon that happens, and I'm hoping it's happening amongst you as well, that you're talking about your own practices, successes, and ways to use things, because it is an incredible process. And I can tell you already this weekend, I've learned as much as I've tried to teach. And it's an amazing process that I hope that you all are both gleaning and can contribute to amongst it. And again, with the breakouts and other things at Sunshine, it's that much more that you're going to interact. So without prolonging that any further, let's start with the slides. So the microbiome, which we've referred to a few times, right? We've almost used the word so much this weekend that it's seeming like old hat. And what I want to remind you of is that it is not old hat. It is a brand new information. How many in this room have heard the term microbiota, metagenomics, or microbiome before coming here today? All right, so the good news is more of you have heard about fecal transplants. But the microbiome is an amazing structure. And I'm hoping that you're connecting the fact that the microbiome, your GALT, which is your immune system in the stomach, your second brain, your intestines, right, leaky gut, they are so interconnected to what kind of issues? I'm not going to use the word disease, but what issues did Dr. Pompa hit on just before me? Autoimmune, okay? Autoimmune. I can't think of anybody in this room that doesn't either know, deal with, or learn about autoimmune, because they are all out of control. For the first time in recorded history, they are out of control. They are literally epidemics. The last time we saw such things, we were talking plague, we were talking HIV, we were talking these acute scenarios, which many, research is well adapted to acute scenarios, isn't it? Right, look what we did with antibiotics. Right, before antibiotics, we put a lot of effort into the acute situation of an infection. Energy, effort, and we discovered antibiotics and we went after it and we tried to eradicate them. Vaccines were another attempt at an acute scenario. Guess why everybody in that model is so confused right now? Do you think diabetes is acute? Do you think autism is acute? Does it come and go? No, it's a chronic. It's chronic inflammation. It is changing lives forever, right? An infection, even if you don't treat an infection, even if the people that survived the plague, they got sick, severely sick, but guess what happened? They recovered, right? There's a whole group of descendants in England that their town is being studied because they were all infected by the plague, but none of them actually got sick from it. They have either a unique microbiome, unique epigenetics, maybe some genetics involved, but they recovered. Or if they did get sick, they recovered just fine. That's an acute situation that your body can come back from. These chronic illnesses, these chronic autoimmune systems are not the same scenario. And that's why everybody in the old model is scratching their head, is beating their head against the wall. They're a terrible, terrible thing that we are on the cusp of moving forward, inching forward every time. The stuff Dr. Pompa talked about, Dr. Tips, hopefully the stuff I'm presenting is allowing you to make these new connections in your brain and in your practice. Say, wait a second, this is a holistic approach. You do not take an antibiotic to fix autism, do you? It is not an acute solution. Changing the mindset entirely. The microbiome is critical to that. So, Why is it? It's not only an interesting piece for us. There's been over $170 million spent just in the United States alone studying it. Now keep in mind, most of that's basic research. We get to take advantage of that information because they're understanding what's going on in the microbiome, because they want to see what's it like in this population, young, old, infants, the elderly, and there's a lot of patterns that start to emerge. It's how we interpret those patterns that will make the difference in health. Otherwise, it's just information. You know, look at an echocardiogram. Look at your plates, right? You need to know how to interpret information in order to make it utilizable. That's the piece that you guys are getting. All right, so what about it? Well, we know that it's a critical part of our immunity, communicates directly with our own cells. Digestive health is, of course, a big issue. And then we have this dysbiosis and other issues. This is a fun picture. This is actually taken right out of the Microbiome Project literature. If you look it up, you can see how many people have intellectually gotten involved in the project, and it's pretty phenomenal. Now, what have we learned about it? Well, we used to think of our bugs, and I mentioned this a little bit yesterday, and Dr. Pomp has also done a really good job, and Jack and Dr. Barston. It is a new paradigm. And it's a new paradigm because bacteria used to be just these single-cell organisms that we knew existed. Sometimes they were a nuisance, sometimes they weren't. That really has nothing to do with it anymore. Yeah, they're still single-cell organisms, but in your microbiome, they are not a single-celled organism, are they? They are an entire organ. They do not function autonomously. They function as a group. They can win the war with you on things like autism, as Dr. Pompa mentioned, because they function as a group. One of the issues that plagues the people that have suffered from autism, especially in Utah, you know, sadly that's a horrible statistic, I hate to admit it, is that the microbiome and the body, and the tissues in the body, don't seem to deal well with mercury in point of fact because of some low glutathione production, right? Now glutathione, many of you guys have probably studied glutathione. Dr. Pomp has been talking a lot about glutathione. We have some reduced glutathione in there. And I've received a few calls over the year, and they say, what happens to the reduced glutathione? Well, for the most part, if it's not the acetyl, which is in the product, but the other one, just reduced, pure reduced glutathione, you're right, it doesn't make it into the bloodstream. And I don't know if anybody in the room has been on a phone call with me when I've said that, but my next part of the answer is this. That's not why it's there. It's there because the microbiome, or the colonies that capsule dissolves into, the gut, those probiotics, guess what they do with it? They use it for the same purposes, to chelate heavy metals. We talked about one yesterday. If the heavy metal gets in there, doesn't get chelated, it's going to be taken up by that dosulfuribio and be converted into methylmercury. It has a purpose, even in the intestine, as reduced glutathione. I don't want you to overlook that fact. Just because someone said the blood levels don't rise, that doesn't make it a bad thing. It is good for the microbiome. Because they don't produce it, right? Not many bacteria produce glutathione. But you need to remove the heavy metals from your microbiome, otherwise, inevitably, they're going to go right through. Okay, so that's just a little side note. Commensal versus pathogens. You know, we've always separated them, and now they're one and the same. I'll come back to this because many of you had interesting plates today. And what you used to maybe think of as a pathogen lives with you, and it lives with you in harmony. So don't stress about that. Terrain and toxins, critical factors in this microbiome. Again, there's this amazing justification of our world, this industry, these kinds of, you doctors, you clinicians, LACs, all of you have been working on these premises, right? These fundamental rules that this research is essentially validating, right? Anybody that practices LAC and Ayurveda, I think I said this yesterday, I'm going to say it again. If you look and take home some of the things I'm mentioning here, like herbalomics, you will be able to validate some of the things you were taught as a traditional Chinese medicine and an Ayurvedic medicine person. You know why? Because they're now no longer just fundamental things that you got results with. The data is demonstrating that they're validated. That you will start, people, if they're smart, people in that discipline that are teaching and pushing that discipline forward, they will look at some of this data and say, guess what? That 5,000-year-old practice, here's why it works. We no longer just need to know empirically it works. Here's why. It's actually preceding the science. The science is going backwards and supporting and validating these techniques that we've known about. So who knows what's to come for us, right? It's amazing. Prebiotics, of course, and then colonization. We talked a lot about that. So here's a little deal to access the millions of genes. So, and I know Dr. Pompa and Jack spoke about this. It is critical. It is critical because I'm going to hand around this sheet right now. And I want you to look at these. I'm going to send one down this side, one down this side. Hopefully they'll converge and come down the middle because I only photocopied 2. I want you to look at this sheet because I want you to see the diversity, not only the diversity of the bacteria, okay, that colonize the gastrointestinal tract, but I want you to look at what they make and what they do for you. How many compounds, how many wonderful things they contribute. And the only way to get all of these things, and this is just a short list, by the way, is because they have 3 million genes that you don't. There is no way you would get this in your diet, in your world. The closest you can come is colonizing and using fermented foods and getting those probiotics in you. Because what they make is incredulous. It's an amazing number of things they make, and they speak directly to every tissue. So read this. Take a quick gander because you got to pass it back throughout my talk and let people take a look. But this is just a sampling of the amazing things they can create. I read that. I'm aware of probiotics. I'm aware of growing these things on plates. I'm aware of the natural products they produce, and I have been for 25 years. It's always been fascinating to me. But the microbiome research has demonstrated that it is, even microbiologists have been studying the tip of the iceberg, less than the tip of the iceberg. The Microbiome Project has opened up this world of, we had no idea. We knew they existed on meteors. We knew they existed in nuclear reactors, and great, that's novel. We had no idea the impact they had on our world. And every aspect of these 3 million genes gives you that arsenal. You make so many more things. I'll pass out some plates here in a minute too, after I describe them, that they make so many things. We don't need all this synthetic junk that we've decided is good for us. There are so many things you can get naturally that can't be touched by anything that a company can produce synthetically. I don't want to pick on, you get the gist. This is real quick. It gives you some count. All of us have used a slide like this, so you can see that there's a huge load. So don't be surprised on your plates if you have a lot of bugs, because they are everywhere. That's the first not shocking thing. So those of you that looked at your plates and said, whoa, don't be shocked. They are everywhere. You can see these counts. If anybody looks at these numbers, 10 to the 4 is 10,000, etc. So there's a lot there. All right. So nutrient conversion. So here's some of the selective advantages for us. So these are the run-of-the-mill advantages. We're going to talk a little bit more in depth today about the novel or the unique advantages. But we've got novel enzymes. We've got secondary metabolites. We've got defense. They're a biofilm. Okay, anybody heard of biofilms? Yeah? Generally speaking, we think of a biofilm as a negative, correct? And indeed it can be. Indeed it can be absolutely dangerous. But in this case, your body has a biofilm that's absolutely positive. These probiotic biofilms can be your best defense against everything. Really quick, I'm going to draw a thing on the board. I don't know if this will really translate well for you, but it's my little deal. So, in order to get a healthy human thriving, so we don't want survival, we want thriving, right? We want a thriving life, not just a mediocre surviving life. That is not our goal here. So to get here, to get to this thriving, we have us, right? So we have things like food, we have things like probiotics, nutrients, we have herbalomics. All of this has to go through the microbiome to get here. So no matter what you put in your body, even when you inhale it, even when you fecal transplant it, no matter what you do, even if you head towards the skin, it has to go through your microbiome to make it work. I don't care what it is. That's what it has to do. Because of that, we can no longer ignore this biofilm. Because it can turn things into good things, it can turn things into bad things. I'll have a few bad ones later. Methylmercury was my example yesterday. We have to take this into consideration. Because on this side of the equation, it's too late if you don't consider this. All right, moving forward. Cool. So novel enzymes, this actually comes into play when we loved Sarika's talk, right? Sarika. Now, I wouldn't necessarily I thought of Sarika as more practical. I don't want her feces right now. Maybe later. I'll put my name on the list. But what I do want, her kid's feces, no. What I do want from Sarika, though, is I would love her to move to Utah right next door to me. Right? I mean, come on. Not only do I have, it's way beyond the fecal transplant. Like, that's a one gift. That's a gift. I want the gift that keeps on giving. And that would be her neighbor with a gate, right? And I would be like, I'm home from work. Let's go over to Sarika's house. Come on, everybody. The whole family. I mean, we would all be happy. That's my wish for Sarika, that she moves right next door to me. Yeah, and if I needed her poop again, I could do a family poop thing, right? Yeah. Hey, it's only 4 syringes. That's all we need. But based on her stuff, I would love to remind you guys that all these things down here below, these are all part of the idea of fermentation and probiotics. So the MBC, the ABC, and all these fermented products, this is what they give you, and you can't touch it any other way. You just can't. I don't care how you look at it. And don't go eat dirt either, because there are some bad guys in dirt, but fermentation is a wonderful, wonderful thing for you to do. And you get all those other things, okay? So that's absolutely necessary. And like she said, some of the fun things about it is all these things that are produced are things we haven't even touched. We look at microbial and fermentation metabolites, and every time you look into it, we find something new. We find new antibiotics. We find new enzymes. We find new things that are converted that are absolutely wonderful. And keep in mind, these guys, I don't mean to sound graphic again, but these bacteria and these yeast, and it's a combination of them, the bacteria are having sex, they're passing DNA back and forth, and they can do it at an alarming rate. You can take advantage of that. Your body takes advantage of that. Okay? All right. Moving on. All right, so here we go. So the diversity, we talked about that. Again, this one's kind of a re-deal, but we're talking about a lot of information here. But really quickly, I want to point out that you've got streptococci, lactobacilli, gram-negative bacilli, Prevotella. Look at all these names, because those are on that sheet of paper I ran around to you. And they fall into 2 main categories. Is what we call bacteroides and firmicutes. Okay, those are the 2 main categories. Now, we didn't even know that before the Microbiome Project. Right? We learned that throughout. There's been over 5,000 people looked at, which means probably 40,000 pieces of feces have been studied. It kind of sounds weird, doesn't it? That we've studied that much feces, but there's a lot there, and they've biopsied. They look at every age group, and we'll get into that. But what we found is all these deals. I think it was it Dr. Pomp, I believe, that talked about Helicobacter pylori? It actually is a probiotic, believe it or not. There are instances where when you have dysbiosis that it can cause problems, and I think Sarika mentioned the low hydrochloric, hypohydrochloremia. That one is a problem. But so many people carry H. pylori, and when you remove it, right, when let's say someone goes to their doctor, they hear they've got it, they don't have symptoms, they don't have an ulcer, they don't have gastric cancer, but people were going in and getting tested for it because it was thought of as a pathogen right after its discovery, right? And that actually was probably the first discovery of the microbiome that was unusual, because the guy that discovered it, an Australian researcher, he presented that at a microbiological conference, and guess what they told him? They didn't believe him. They kicked him off the stage. They're like, get out of here, you're crazy. There are no bacteria in the stomach. They did. I mean, I'm not even exaggerating. And then he went back home, and like all good scientists, he ate it. And he gave himself ulcers. And he went to the doctor, and he did all the tests. He was his own experiment, right? How many of us have done that? Right? Yeah, come on. I know you guys have. Hey, I know Sarika has because she ferments food, right? That's that's an ongoing experiment. Well, that happened, and now we know H. pylori, and it's actually predominantly higher in populations than we ever suspected, living harmoniously for many of them. So again, we got to look at the bigger picture. Why would it become a problem for some people? I speculate because there's other dysbiosis, there's other inflammatory issues going on, and now it becomes an opportunistic problem, not that it was always a bad guy. And your immune system, depending on your individuality. Some of the other things you got to pay attention to. Lactobacilli, you're probably all very, very well versed in that. I want to point out something too. Gram-negative versus gram-positive. That is a nomenclature used by microbiologists. The only thing you need to know is if you run tests or if you get patients that are not having success through the traditional healthcare guys, that route, and they come to you and they have data, gram-negative can be your friend, but gram-negative that produce LPS, lipopolysaccharides, they are not your friend. They can, in many people, exacerbate the inflammatory response, the leaky gut, they're connected, and so are a lot of the autoimmune subsequent to that. It is a marker that infuriates your immune system, okay? Because it's a trigger saying, we've now got these bugs in our bloodstream, and it raises it to that, code red, I guess? Okay, so it's just a consideration if you get this on your data. Bifidobacteria, these are just some fun. Now, this methanogens, there is a very unusual way to test for, see this methanogens down there? Guess why they call them that? They produce methane. That is the bacteria that teenagers love to take advantage of, especially if they have YouTube. Because guess what they do? They light their toots on fire. And it makes that just, that little burst of flame. Those are the methanogens. Now, when you have overgrowth or if you have dysbiosis, that changes. So it's another kind of unusual test. I won't actually push you down that direction necessarily. Yeah. Okay, so you are what you eat. And Nicole was saying something, I'm not sure if you actually got to say this before, but what we don't want you is quick and easy. And what was the other thing, Nicole? She had a pretty good, clever saying.
Am I on?
When she saw my slide.
I said, if you are what you eat, then don't, and you've probably seen this on Facebook, then don't be cheap, fast, and easy.
Cheap, fast, and easy. Don't be that person, right? Don't be cheap, fast, and easy. I thought it was pretty clever after she saw this on my slide. No, we don't. We want to be deliberate, thoughtful, and healthy. So, we are what we eat. So I just pulled, this data is abundant now because of the Microbiome Project. And the researchers are actually quite clever. They're looking at a lot of diversities. So if you look at this one right here, this is Burkina Faso in Africa. This is Florence, Italy. This is a really rural place in Africa. Okay, it's actually a rural, very, very ancient place in Africa. And so the kids they were testing, and they talk about it in the paper more about the details of it, which is down here if you want to go look for it. That's the reference at the bottom. But look at the breakdown. So what I want to point out here is, I've just mentioned the names, but let's break down this first. So this red right here, that's the firmicutes, right? And here's the firmicutes. This is Bacteroides, and the blue is Bacteroides. Look at the difference between the two. Huge difference, right? Now, I can tell you that this young man versus this young man, they looked at their diet, and they were dramatically different in ways. This young man was eating a lot of ancient grains, by the way, because he didn't have access to a lot of meat. This young man had a significant amount of meat in their diet. So that was their first hypothesis, that those changes in what the bacteria is getting daily will adapt to what it's getting. And it does, in fact, because we've shown that now with big transplants. We've shown that with altering diets of twins, taking the twins that start off the same, not the ones that are different, or even populations are the same, and feeding them separately and watching the microbiome change and adapt. And it's clearly happening. In some cases, you can run it the wrong way, especially if you're cheap and easy. You get growth, you promote growth of the wrong bugs in almost all cases. So in a lot of the cases I'm going to talk about today, you can have variations. If all of us were to get tested, we would have subtle variations. Some might have more extreme variations depending on whether you're in a climate that you have access. For example, I think Sarika's would be very different from some of ours because of her natural diet, the diet she's eating daily. But we all would have some subtle differences. However, it's clear that when you look at the people that are harboring the autoimmune dysfunction, and we'll get into more of that, have this dysbiosis, or these variations, like on this chart, that are going the wrong direction. You can have healthy variations and you can have unhealthy variations. And IBS, IBD, Crohn's, celiac, those variations are becoming more and more dramatically identifiable. And a lot of it has the diet. I mean, I don't know how many times you've heard us, I've heard you guys talk about how diet and how nutrition, how herbalomics can play a positive role. But if your patients have none of that in their world, then their microbiome is struggling to keep them healthy. And it wants to, right? It's self-preserving. It fluctuates. It ebbs and flows in these different organisms because it's trying really hard to do that. So let's keep going. All right, so normal gut flora represented by 6 main phyla. I won't bore you with this too much, but the Bacteroides, Firmicutes I told you about. Lactobacilli are part of that, actinobacteria. Now some of these, like proteobacteria and fusobacteria, these are on your skin. Those ones you guys are going to see, or in your mouth. Enterobacteria, if you guys did the plates, those are the bacteria that live. They're called enteric bacteria. They would cover E. coli, Salmonella, Shigella. Sometimes a problem, sometimes not. Okay, so not only is that a mainline topic for us, but it's been in dozens of major publications. It should be hitting the mainstream here shortly. And we want to stay ahead of the curve, because as soon as it hits the mainstream, you're going to start getting questions from your patients, clearly. We talked about Google yesterday. It's starting to pop up in other areas like Google, etc. And you're going to start getting questions from your patients about, hey, what is my microbiota? What is going on? Why is this important? And having you guys prepared for that is important, because there are absolutely tools that we're offering in education today. We want to change the way we think, absolutely. Again, I can't emphasize enough that this micro world is critical to our autoimmune issues. It's the centerpiece. And I don't want to underplay all of the other things we have in our world, the herbalomics and all of our formulas in the food, etc., and exercise. But the microbiome is where that has to go through to get there. No matter what you put in there, the microbiome can alter it in a number of ways. It has to go through that. That is a group of cells that have to be dealt with. Can't not be ignored. It's like trying to get something into the brain. You have to go through the blood and the blood-brain barrier. That is a fact. Now we have to go through the microbiome. And we want to go through it with what helps, with what works. Okay. Let's talk about a little bit of this. This is kind of fun. How many of you have children? Okay, how many of you, and you know children, you may take care of children. It's clear among our world that the younger you can start things that we teach, the better. I know that I don't mean to scare those of us that are older. We can still help us, absolutely. But the young people have the most to gain from all the things that we do in our world. The herbalomics, the nutrigenetics, the epigenetics, etc. They have the most to gain. They have the most to gain from this as well. Because at birth, you're born with approximately 100 species. And that's something that it's still out there. Now, there's only been a few publications that say that they've found it in the first test of the baby before it's even had any external food, no colostrum, no breast, no formula. So my guess is we may find it for real, that's a real data set, as opposed to being sterile. But more importantly, this data means that we have very few. We start out very short. And there's another study that took infants from vaginal births and infants from cesarean sections. Do you think they're different? Yeah. Do you guys ever recommend when you're talking to pregnant mothers which way to go? I can't imagine anybody here recommends cesarean, right? Now, unless it's a health crisis and it's an emergency and there's no choice. But the problem is, not the problem, but what we found from a microbiome perspective is that the cesarean, guess what their microbiome, intestinal, is made up of? Anybody want to take a guess? It's a tough one. I didn't know the answer. The skin microorganisms that the mom carried. Interesting, isn't it? Now, the other babies that are born vaginally have more or less the same species that the mom does enterically. So it's a very interesting phenomenon. We don't know what the implications are long term, but we know that it takes longer for those infants to gather the steam, because you go from 100 at birth and then you gradually go to 700 in adolescence, different species. So there's this evolution, there's this transition to lay this landscape of your microbiome. That's critical, because once they're there, and the reason we're here is that we want to alter that. Right? In many cases, especially when you have autoimmune and these others, we don't want you to keep what you got when what you got is not working. Right? We in agreement with that? When it's not working, when you have these issues, the microbiome is central to that. You've got to change it. Changing it is a little bit of a task. When they're little, you can put in play the right things. So whether you see pregnant women, whether you see young kids, even pre-20s, right, adolescents, they have the ability to evolve way more quickly because they're still adapting. And interestingly, when you're laying that landscape, when you're creating that evolving gut for them, diet, nutrition, herbalomics, epigenetics, nutrigenetics, they all play a critical role in those people, as well as the adults that we want to change. We'll get back to that. But guess what else is happening during that period of time that seemed to be correlated? Remember I mentioned yesterday we have the HPA axis that seems to evolve with the microbiome? It's true. Now they're watching brain cells. You're putting in about 250 million brain cells a year or something like that as they're getting older. That seems to be correlated to that same transition. The time it takes for the microbiome to evolve is the same time that you've got these other tissues maturing in these kids. Right? They're timed similarly. You don't get the microbiome and then 5 years later get your other tissues that mature, the brain, the liver, they're happening together. It's a coordinated effort. And the reason for that is the microbiome is dictating so much of what's going on inside the body. And you may have stunted growth in certain areas because the microbiome is dysbiosis in those kids. And it doesn't know what to do. It might be trying to send the right information, whether it's through the endocrine system or whether it's through the nervous system, to those tissues as they're maturing. Do you guys see how critical this is? And this is so critical even in us as we age. That link cannot be disconnected anymore. We can't just treat the tissue without its context. All right, so moving on. So we got at weaning, you've got 700 species. Sorry, I said older, but 700 species at weaning. A lot of that comes, of course, from mom and the environment. Now, the environment is critical because a lot of people, well, I know some personally, they have their baby, don't touch my baby. Have you washed your hands? Now that's true if they're carrying, they've got MPV, MPV or something. They're sick and that, but that's different. But they're getting their microbiome from their loved ones while they're also getting it from mom. And that's a critical piece because the hygiene hypothesis runs us in the wrong direction completely. Okay? Now, at adult, you got about 1,000 species. That varies among us. And it changes with environment. Like you saw that young man in Africa versus Italy, American. And then now we complex that with the idea of all of the other environmental factors. Toxins, biotoxins, xenotoxins. What else did Dr. Pompa tell you about? Stresses, external stresses, internal stresses. They all forge a change in the microbiome. Okay, so what does it do? Well, before I get back to some of the changing, these are the things it does. These are the proven things it does. These isn't speculative anymore. Right? This used to be speculative for those forward thinkers. This is now things that we know it does. It has an effect on fat storage. Insulin, type 1 diabetes, type 2 diabetes, type 1.5. Anybody heard of that? Yeah. That's a growing problem. You know why? Because you've got these guys over here that have type 1, and we used to call that what? Juvenile? And why do we call that? Because we suspected that it was, you're kind of born with it, right? Or it's early onset, genetic. And there's some genetic components to it, absolutely. And then you got type 2 that used to be in elderly, unhealthy adults, right? And when I mean elderly, I'm just not talking old people, but I'm talking more aged, right? Wise people. 1.5 now is the bridge. And type 2 is moved all the way down into young people. Okay, now, what about that story makes any sense to you in terms of we're going the right direction? Okay? Nothing. You've got type 1, which was fairly rare, and you used to think you'd only get it if your parents had drawn the short stick on genes, right? Like, my parents don't have it, I'm not going to have it. Great. Don't need to worry. Give me that hamburger. Type 2 now was just that guy that didn't, that was always sitting in front of the couch watching baseball. I'm not picking on baseball, mind you. But he had, hot dog, hamburger, big jar of Coke. It was on a tray, and he never moved, right? He just kind of leaned over. His hands didn't work, There's a cartoon that came out a long time ago. I think it's a Disney cartoon where we escaped the planet because we ruined it. And so they went on this big cruise ship into space. You guys ever remember this? Wall-E, thank you. I don't remember the name. Do you remember the people on Wall-E? Was that not the scary, forward-looking situation? Seriously, they were all sitting in chairs. They hadn't forgotten to walk. They were huge. And food was like squeezed into their mouth from a tube, right? As it was made by a robot behind the door or whatever. I mean, if that is any indication of how people think or how it's been moving, we have a big job to do. But back to 1.5. So now we have to have a new 1.5. Why? Because we're making so many terrible decisions and we're doing things so incorrectly, we have to have a new disease because we have a new set of problems that we've created, right? It's a combination of the two. There's an autoimmune component and there's a insulin-resistant component. Now next we're going to hear that you can go from one to the other. Now that's going to be the next phase, right? You go from something that was essentially a insulin resistance and now you've taken it all the way to the autoimmune spectrum. We are facing epidemics of really scary proportions. Not even just in terms of how many people we're hitting, but how complicated they're becoming, right? They're no longer just that simple fix that people used to like to think they can do. All right, so sorry, I went off on a little tangent. Tissue lipid composition. They control how lipids are formed. The messages coming from microbiome can tell your eyes and tell your other tissues where to put certain lipids. When you eat a fish oil versus an omega-6 versus an omega-9 versus a small chain fatty acid, the microbiome directs some of that tissue composition and control. Wow. Vista, right? When we feed Vista, we want the microbiome to be happy before you eat Vista, don't you? Or at least during those phases. Because if not, you're going to have to work on that patient a little bit longer in order to get that Vista to where it needs to go. Or the MORS, or the E-energy. Right? If you have a dysbiosis, that those organisms are highly consumptive, and there are organisms that are highly consumptive, when you subject those organisms with all these wonderful nutrients, do you think they're going to let them go back into you? No. No, those little microorganisms, the guys sitting on the chair, waiting for you to just keep it coming, right? No, that is a very, very scary scenario. Some of the other fun stuff that we talked about, behavior, motor activity, going back to what Dr. Pompa said, not only is the gut intricately linked to mental health stability, it's important for these things going from just an instability all the way to autoimmune. All right? So I can't emphasize that enough. This is kind of a fun deal because they touch everything in your body. They absolutely touch everything. Periodontitis, energy metabolism, it's all there. It's all there. If we ignored the microbiome, we're missing the biggest piece of the puzzle for intracellular healing. Okay? All right, so this slide you saw yesterday, I wanted to give it back to you again. Just now that you've heard more about the microbiome, this little place interacts with so many different systems. You've got the spinal cord system, you've got the adrenic system, you've got the vagus nerve, you've got the endocrine system. It's hardwired to everything you do. Hardwired. All right, moving on. So external influences, we've got diet, antibiotics, pathogens, immune system, genetic background. We can't forget that. I think someone over here mentioned that yesterday. And then the GI physiology, transit secretion. So these are all the external influences. Clearly, the environment changes everything. Okay, so let's look at this. Here is one of the studies that Dr. Gordon, I believe, published. And I can give you a number of these. This one is based on inflammatory bowel disease, type 2 diabetes, and necrotizing enterocolitis, I believe. I can't see that at the bottom. But here's 3. Okay, I can give you these for every autoimmune. There is a graphic in the literature for every autoimmune disease and other chronic issues related. And this graphic says what? Well, here's the healthy control. And what we have here is the firmicutes right here in purple, Bacteroides here in the lighter, and then we have a combination of these other ones. Look what happens when you go from the healthy control to these guys. Look how things change. Now, obviously, in fecal transplants and this future pacing product that Dr. Pompa talked about is in this area right here, Bacteroides. Right? Healthy controls have a significant amount. Now, this is a totally different issue. I'm not going to talk about this today, but this is a really unique situation. But Bacteroides is clearly helpful. We don't give Bacteroides. We don't have the ability. So what you get from your diet is the only way to do it at this point in time. Or, like our band, fecal transplants. That's how you get it. Now, if you don't want to have the deep enema, there isn't an oral option, but I can tell you, because as Dr. Pompa and I have been talking about this over the last 6 months or year, whatever it's been, I can't even remember now. Time flies so fast. That there are a lot of studies of oral fecal transplants. I know it sounds a little gross. The veterinarians are doing it, and humans have done it. They've done it for some humans. Okay? The research has shown. Now, you get a subtle improvement over the fecal transplant through the enema. But it's subtle, which is really encouraging, because if we can get those same organisms in a capsule, then the oral route is only like 2% less efficient of getting those bacteria recolonizing. All the same group. So they were studying a group of IBS. It was the C. diff. It was the C. diff that they did both oral and fecal. And the subjects had now get this, they had a 92% success rate with no recidivism, which means they never got it again. It was gone, and they followed them for years. That's unheard of with C. diff. C. diff is a chronic, reoccurring little bugger. Now, the oral, they had 85%, so really good percent, even with oral. So that's very encouraging for people that will never do the fecal transplants, or it's hard for you to do in the office, whereas oral would be much easier. So I was very encouraged by that data, personally. And you can be too, because as you eat the capsules you're going to be eating, the secret is that they contain Sarika's feces. No, I'm kidding. I'm just kidding. I'm not just kidding. They don't. But would you be mad if they did? All right, that's good then. Maybe next time. Poor Sarika. I hope she's taking this lightheartedly, because we love her. We love her to death. All right, but the good news is what Pompa suggested is possible in the future is possible. That we will hopefully have that come that day. That we are giving you something that nobody else can give you through that route. But very great data. We're going to use this data to kind of drive some of our research. Here's a couple of things that he did. So microbiome analysis of obese patients have more firmicutes, just like I said, fewer Bacteroides, than thin patients. And guess what they did? They reversed and gave the thin patients and swapped the ratio. And guess what happened to the thin patients? They started to gain weight. Right? Not a good sign. PEG treatment. Now, this is not a good thing to do, but it was a good research piece. And I really don't know, how they talked these patients into doing this, because it's pretty harsh. But if you take a PEG, which is polyethylene glycol, and you flood the colon with it and the lower intestine, you literally are stripping it of everything, right? It's kind of a solvent. Now, it's not deadly, obviously, but it's going to remove everything. And what that did, that allowed for these guys to then transplant from thin patients, and obese resulted in weight loss. How cool is that? Right? Now, there's two little notes I took from this. One, it's probably a good idea to move it down a little bit. Take down what you've got. from those people, right? You can't just add to what's there. You might have to remove a little first. And that's part of the protocoling that all the doctors we're working with have suggested. Danny, Sarika, Barston, and Jack. And it's not a novel concept, but again, the concept precedes the data, right? Just like I said about LAC. This is now data. The concept was out there prior to. So isn't that a wonderful, miraculous thing that this industry can do? That people in your position are doing? You guys don't get the accolades. I mean, there are ideas that have come from this group, a group like yourself, that are forward thinking. But you guys are out there just keeping pushing forward. You're not stopping to go and get the prize, right? To walk up on stage. Maybe we should have our own little prize for forward thinking ideas, right? Yeah, maybe we should create something. Let's keep that in mind for next time. Don't let me forget. Okay, so another one was a malnutrition study. Was caused by dysbiosis. So microbes taken from one twin transplanted into the sterile mice resulted in malnutrition. They took it from humans, actually, and had sterile mice and found that they could induce malnutrition in mice from the microbiome of the malnutritioned human. Right? That's all they did. They still gave it all the nutrition it needed. So what's happening? Nutrition, bad microbiome, bad health. Pretty fun. All right. So we want to change the behavior, we want to change the microbiota, we want to change the gut-brain axis. A resounding yes, we do, especially when we're talking about existing issues that people have. All right, here we go on a specific outtake of obesity. So clearly obesity, and again, we talk about plateaus. You might be getting patients a little bit up to here, and then they plateau, and you can't get them over that next step, right? You've tried lots of things. You've done the cellulars. You might be doing the GA adrenal, dealing with the thyroid, right? And you're like, wow, my thyroid seems to be getting better. The data's demonstrating it, but they're still fighting this problem right here. Well, here's just a little graphic. I know there's a lot there. You can study it later. I know you're going to read this tonight. Adipose tissue. It is modulated. Remember the nutrigenomic maps I've been drawing you where you have signals coming into membranes. That membrane signal gets translated through the membrane in the nucleus. The DNA gets activated, sends out a messenger RNA, which becomes a protein, which becomes a signal, which becomes the activity that happens, right? That process is in everything. That is life's process. Well, here it is. You've got lipopolysaccharides, which come from bad bacteria. They get through the tight junction that's broken. Go figure. We know that happens in a lot of our patients. They trigger on the cells a process that tells the cells to take up fat and create adipose tissue. So your microbiome is driving fatty acid depositing in a way that you don't want it to do, right? One other mechanism that you got to be careful of. This is all the details of it, and it's unrelated to food intake. Even when they studied people that had lower than normal caloric intake, if this is going on, you're going to get this. So it's pretty impressive data. Again, it talks to the bigger picture of why we want this to happen the way it is. All right, next slide. Another quick one, the insulin resistance. Okay, there's a number of ways to get insulin resistance, and we've talked about it from cellular healing perspective. We've talked about it from toxins perspective, and now we're going to talk about it from the microbiome perspective. Because remember, insulin resistance, like thyroid, can be a problem with the insulin hormone or the receptor or glucose receptors based on mitochondria. I won't bore you with that too much of that, but if you have damaged mitochondria, it immediately sends a signal to the cell surface saying, don't take any more sugar in. Well, if you have mitochondrial diseases, this is a mechanism, this is an herbalomic mechanism, right? And the mitochondria is damaged, it tells the cell, no more sugar, we're in trouble, we can't take any more, so don't let it in. It shuts down the glucose transport proteins. Insulin's receptor is still working fine, but insulin's binding. The brain's saying, come on, man, take the dang glucose up. And the cell's saying, no, man, I got damaged mitochondria. I can't do it. That's one mechanism. Okay, so again, we're talking about a holistic approach. Another mechanism, as Dr. Pump talks about all the time, is the insulin receptors are damaged. The membrane is damaged. Different scenario. You damage that membrane, that insulin binds to that receptor, the signal doesn't get translated, no sugar comes in, sugar remains in the blood, you start making more insulin. Pretty soon, those receptors become negated, essentially. All right, so immune sensing, we talked about that at length. I just wanted to give you guys another way. So again, this is the pathway of things that happen. This is a flow chart, so to speak, to get down to these problems, right? And what happens is because of the connectivity with these T helper cells, and these are thought of a lot as innate and adaptive immunity, when you start up here with all these dysbiotic sensing, the leaky gut, you trigger these T helper cells to head down the wrong path. And now we have autoimmune. So again, another map to show you you're going the wrong direction. All right, so without our microbiota, we lose the innate and adaptive immunity. It really gets confused. It really does. As I mentioned, it's sensing your gut all the time. All the time. We're talking about the quorum sensing. That was that little, I don't know if you remember, but it's a community that it interacts with us and we interact with it. It's just like the nervous system. You're talking to your microbiota all the time. And of course, the hygiene hypothesis is gaining a lot of energy now that we have this information. The neurotransmitters as well. So now it seems obvious, because how many people have recommended, I don't care whether you watch TV, you read books, you see the videos, the rage of these diets and exercise. Well, one of the problems is you've got, when you throw somebody into a diet or an exercise and they've got this problem, you're exacerbating it. You're going the wrong direction. You need to hit them with the microbiota, and that's in the package I'll get to really quick. You got to start there, because if you just say, oh, exercise and diet, you're going to miss the whole boat.